cute little champions, I welcome you all to mock test 10 of our NEET Premier League. And today, for our mock test, I have chosen this chapter, that is Biotechnology, Principle and Processes. So whatever previous year questions were there, 30 of them, I have chosen, I have put it over here. So you can have a quick practice. Remember, they are all previous year questions, so they will give you a good idea. Now, before starting the video, I want to tell you, have you tried solving the questions by yourself? If you have tried, then only watch the video, or else you can just wait for some time, try it out and then come watch the video. Okay, this video is for making sure that your answers are correct. Just for you to tally your answers, I am making this particular video, and also to explain in some of the other ways, so that some children know they learn by solving questions. So that is also a good method, but yes, obviously you have to try for yourself. Okay, chalo. So let's start. And children, please give yourself +4 mark for all the correct answers and -1 for all the negative, all the wrong answers, because you know, right, NEET marking schemes are like that, and we have to practice it. Chalo. So which one of the following is a case of wrong matching? Okay, this came in NEET 2012. Somatic hybridization means fusion of two diverse cells. We want to find what wrong matching. So please underline wrong over there, because sometimes people get confused. What are you searching for? So yes, we have to look for long thing. So somatic hybridization means fusion of two diverse cells. It's fine. Two diverse cells, you are hybridizing it. That means somatic hybridization. Okay. Now going ahead, vector DNA. Vector DNA is the site for tRNA synthesis. That is kind of wrong. Okay, vector DNA is basically, for example, I have Salmonella typhimurium, that is a bacteria, and I want to put its plasmid inside E. coli. Okay, inside E. coli, I did some alteration and then I'm putting it. Basically, this particular plasmid which is there, which is belonging to Salmonella typhimurium, is my vector DNA. Is my vector DNA with my gene of interest, and I am putting it in the next organism. So it does not have anything to do with site for tRNA synthesis. No, that's wrong. Vector DNA is responsible for carrying the gene of interest. And this is the gene of interest, children. Okay. Now, micropropagation means in vitro production of plants in large numbers. Yes. Callus is unorganized mass of cells produced in tissue culture. So what happens in tissue culture, children? You have certain cells. You take actively dividing cells. Okay. You take them. You put them in the growth medium. They start dividing, and you have multiple cells now. And this group of cells, you call it a tissue. Basically, you call this as a callus. What do you call this? This is a callus. This unorganized mass of cells is known as callus. Okay, and then callus is later provided with auxin for root growth and for shoot growth and things like that. So different hormones for root and shoot growth. So now this question is a mix of so many chapters. You saw that. Okay, there are so many chapters being involved over here. Now from this chapter, I studied about this and I got to know that this particular statement is wrong, which is why I was able to answer. So sometimes at least a good knowledge in one particular chapter could give you marks. So if just imagine that you do not know about this question at all. Okay, just you didn't know about this vector DNA. Would you be able to answer this? No. Okay, so that is the thing. Now going ahead to the next question, which one of the following techniques made it possible to genetically engineer living organisms? So genetically engineer living organisms means I am going to alter their DNA and put my DNA, not my DNA, basically the gene of interest. Gene of interest is nothing but a segment of DNA, children. It is a segment of DNA, and where I am going to put the segment of DNA? In the plasmid or whatever I have. In the plasmid, or probably a normal DNA strand that I have, whatever it is, I am putting it there. Okay, what I am putting? Gene of interest I am putting. Now, this method of genetic engineering, or this thing, genetic engineering, was made possible because of what? What was there? Recombinant DNA techniques, X-ray diffraction, heavier isotope labeling, hybridization. How was I was able to do genetic engineering in living organisms, children? It was recombinant DNA techniques. It was recombinant DNA techniques where I am using my gene of interest, I am putting in somewhere. So I am doing a recombination. Initially I had a proper plasmid. Now I am recombining it with the gene of interest. Now this is known as recombination. Now whenever I am doing this, this is known as recombination DNA techniques. Okay. Works is again a very easy question. People usually get confused with hybridization and this. Hybridization means you have two plants. One plant has good genes for disease resistance, and one plant gives great fruits. So I fuse them. that is hybridization over there. When I am making them fuse and I am making them grow, whatever is there, okay, I am doing vegetative propagation, all this comes under hybridization. But over here I'm doing nothing which is related to cutting the gene and putting it inside the plasmid. I'm not doing things in molecular basis, which is why this becomes wrong. So when I'm talking about genetic engineering, just understand you're talking about molecular basis, and for that you need recombinant DNA techniques. Works? Next question. Which of the following are used in gene cloning? What do you mean by gene cloning? Basically, you want to create multiple copies of one particular gene. So which of them is utilized over here? This is NEET 2010 question, and this is direct question. The answer will be nucleoids, lomasomes, mesosomes. What is the answer, children? It is plasmids. It is plasmid. What is plasmid? If you are not aware about plasmid, even though I have taught you in the class, it's totally fine for me to tell you. So listen, every other bacteria, prokaryote, no? Basically, they have their own chromosomal DNA, the DNA that codes for protein and everything. Okay, this is what, children? This is the chromosomal DNA. So apart from it, they also have certain extra chromosomal DNA. What is it, children? They also have certain extra chromosomal DNA which is not coding for any proteins or as such, anything like that. And this thing, you call this as a plasmid. Now, how is this used in genetic engineering, ma'am? Basically, I take the plasmid out, okay? I take the plasmid out and then I cut a part of this particular plasmid and I put my gene of interest there. And then I insert it again. They will also copy themselves, just like how the... So chromosomal DNA is obviously replicating whenever you want to make baby bacteria, right? You want to make, you want to divide yourself into two different organisms, okay? Whenever reproduction is happening, at that point of time, DNA is also getting replicated. So it's not like the chromosomal DNA is the only one that gets replicated. Plasmid also gets replicated. So basically, if I had the gene of interest now and I'm putting it back over here, okay, and I'm putting it back over here, in the bacteria, the plasmid will anyways replicate and increase its number. So basically, in a way, am I not cloning my gene of interest also? This is the gene of interest and I am making multiple copies of it. Okay, so the answer for this question becomes plasmid. Cool. Now, next, manipulation of DNA in genetic engineering became possible due to the discovery of what? Manipulation of DNA as in bringing out any changes in the DNA. For example, over here, I had the plasmid DNA. I made a cut and I am putting my gene of interest. This is manipulation of DNA. I am changing the whole structure of the DNA. Now, this became possible when? How did it become possible, children? Is it what was that something that got discovered and all of it became possible? Is it restriction endonuclease, DNA ligase, transcriptase, primase? All of them are enzymes. But we should be focusing on restriction endonucleases. What do you mean by restriction endonucleases? These are the enzyme, okay? These are the enzyme which, for example, this is the DNA and this is the gene of interest. Okay, this particular portion I want to cut it out, okay? So for that, I need an enzyme and this enzyme is known as restriction enzyme. Well, here I see my DNA or gene of interest is within this strand. Okay, so I use a special type of restriction enzyme which is known as restriction endonuclease. Endo as in inside and nuclease as in it is going to cut the nucleic acid. DNA is a nucleic acid, RNA is a nucleic acid. So over here I am cutting my DNA. It is restriction endonuclease. Now for example, my gene of interest is over here somewhere. And 2 is a tip. So at that point of time, what do I use, children? I use restriction exonuclease. It is not within, it is outside. So it will be restriction exonuclease. So manipulation of DNA in genetic engineering became possible due to the discovery of what, children? Restriction endonuclease. This becomes the correct answer. Cool. Now, next question. Identify the wrong statement with regard to restriction enzymes. Okay, how do they work? These enzymes that I told you about, right? Okay, there are different types of restriction enzymes. Now, the question is, what is the actual method of their working? What is wrong? That is something that you have to find out, right? Wrong, right? The wrong statement. So, each restriction enzyme function by inspecting the length of a DNA sequence. Yes, it does inspect the whole length of the DNA sequence, and this is a direct thing from your NCERT. This is a direct line from your NCERT, and children definitely tend to not pay attention to this detail. So, this is a very important thing. Okay. Now, they cut the strand of DNA at palindromic sites. What do you mean by palindromic sites, children? Palindromic sites as in from both the end, if you read it, it will be same. For example, if you talk about Malayalam. From here, if you try to read it, it will be Malayalam. From here also, if you try to read it, it will be Malayalam. So the restriction sites, as in, for example, if this is a DNA, I have uncoiled it, okay, this is a DNA strand, then I say from 5 dash through, this is 5 dash to 3 dash, and this is 3 dash to 5 dash. They are complementary strands, okay. So over here, the site is GAATTC. So here it will be CTTAAG. This is a palindromic site. This is, children, a palindromic site. So over here, if you read, it's same. Here also, if you read, it's same. So basically, for example, I am giving the example of an restriction enzyme which is known as EcoR1. EcoR1. Okay, that's the restriction enzyme. And EcoR1 goes and cuts in this particular sequence. They have a specific sequence. Only if they find this particular sequence, they will be making a cut. Okay, so this is known as a recognition site. What do you call, children? This is known as recognition site of the enzyme. Cool. Now, where are they going to make the cut? In the recognition site, they will also have a perfect area for making a cut. So over here, they are going to cut between G and A. Similarly, over here also, they are going to make a cut between G and A. Okay. Now, you will be left with something like this. You will be left with something like this, where you have AATTC over here, and you have G over here. This is 5 dash to 3 dash, and this is 3 dash to 5 dash. So there is a sticky end. There is an end that is left, which do not have any complementary base pair. Now this is a strand, gene of interest strand. Now I am going to put it somewhere else in the plasmid. In the plasmid also, I make a cut like this, and I find that the complementary base pair of this is over here. So they will stick to each other. I don't know how far you understood, but this is how it works. Okay. So basically, yes, they cut the DNA, they cut the strand of the DNA at palindromic site. So this is a palindromic site. So they will make the cut rightly over here only, nowhere else. Now they are very useful in genetic engineering, children. Yes, they are very much useful in genetic engineering. Obviously, genetic engineering means you are manipulating the DNA, you are making use of the DNA strands for your own good. So yes, you are definitely doing it. Now sticky ends can be joined by using DNA ligases. So this is a sticky end, and I am saying the plasmid also have a sticky end like this. The plasmid where I am going to incorporate stuff. Okay, suppose it also has a sticky end like this. Okay, where the sequence will be what, children? Where the sequence will be GAATT and C. So here the sequence is AATTC. So over here, what will come and bind? This particular strand will come and bind. This particular strand, okay. So over here, if I just erase this, okay. So this guy goes over here. This guy goes over here. What do I have at the end? G. I have G at the end. Now there's a new guy that comes over here. Okay. What do I have at the end over here, children? It is AATTC. Didn't I make the complementary strand? Yes. So basically there was a sticky end over here in the case of plasmid, and there is a sticky end over here in the case of my gene of interest. This is sticky end and this is sticky end of the plasmid, and that's how they join. So basically they are complementary to each other. Basically the strands which comes, they are complementary to each other because the plasmid is also cut with the same enzyme. The plasmid and the gene of interest is cut with the same enzyme because, you know, recognition site should be having the same complementary sticky ends, right? So that's why they are cut with the same enzyme. Now when you want to join them, usually they get joined very easily because they have hydrogen bonding between them. Okay, so you know A binds with T with the help of 2 hydrogen bonds. G binds with C with the help of 3 hydrogen bonds. So this bonding naturally happens. Okay, so it actually does not need DNA ligase. Where do you need DNA ligase? For example, I want to finish the whole stuff. Okay, I want to complete the sticking part. So sticky ends came and it went really well with it. But now I want to ligate this DNA, that is my gene of interest, and the plasmid's DNA. Okay, I want to ligate, I want to bring it together completely without any fault. So for that I need DNA ligase. For that, actually, I use DNA ligase. So sticky ends can be joined by using DNA ligase. That becomes incorrect in some other way, even though it's not completely incorrect. Okay, but yes, if you have to choose one particular statement from this, it should be this. Okay, so DNA ligase is actually used for, sticking down 2 different sets of DNA that we have. Okay, not for sticky ends. Sticky ends can find their complementary base pair and then can easily bind with each other. So this becomes the answer Okay, now that's a tricky question, I'm telling you. Not entirely correct, but that's how the question came. So we have to solve it anyhow. Now choose the correct pair from the following. Ligases join the two DNA molecules. Yes, children. The first question, we studied about DNA ligase right now. So it joined the DNA molecules. It is correct option. Now polymerases break the DNA into fragments. No. Polymerases, for example, I have a DNA strand. I want to make a complementary DNA strand. This is used for replication. So polymerases are for that. Nucleases separate the two strands of DNA. No. Nucleases are for cleaving or making a small cut in the DNA. Okay. Now, or RNA. Exonucleases make cuts at specific position within the DNA. No, children. I told you restriction endonuclease is there, restriction exonuclease is there. So endonuclease makes the cut within the DNA, whereas exonucleases, they cut towards the ends. Okay, so that is the difference. So you should be knowing it. So small details are very much important. This again came in NEET 2020. Cool. So this is the answer. Next question. The specific palindromic sequence which is recognized by EcoR1. I told you there's an enzyme EcoR1. So basically, what is the recognition site for that? It was GAATC. Now TTC. Now please make sure on the side of G you have the 5' and here you have the 3' end. Now if you want to make a palindromic sequence of it, then make 3 and then you have C. C and the complementary strand will be TTAAG 5'. Fine. So this is the strand we are looking for. This is the palindromic sequence, my dear children, and hence A becomes the correct answer. This came in NEET 2020. Cool. Chalo. Going ahead. Question number 8. The sequence that controls the copy number of the linked DNA in the vector is termed. The sequence that controls the copy number of linked DNA, as in, for example, this is a plasmid. How many times this particular plasmid will undergo replication, how many copies will it make, is decided by a specific site. Okay, in the plasmid or in the DNA sequences, that normal DNA sequences, linear DNA that we have, that site is known as ORI site, that is origin of replication. So origin of replication is responsible for initiating replication, children. It is responsible for initiating replication, okay, and also for determining the copy number. For two things you need this. Works? So that is the correct answer. So ORI site is the correct answer. Cool. Now question number 9. In gel electrophoresis, separated DNA fragments can be visualized with the help of. So gel electrophoresis basically is a tube like this, and over here you have the DNA being separated along with the fragment length. On the basis of the fragment length, they are getting separated. So the smaller fragments move really fast, larger fragments move far away. Okay, they move really slow. They move really slow. And where are they moving, children? So you know that DNA is basically negatively charged. Do you know that? DNA is negatively charged. Okay. So negative things will be moving towards positive things, because if you have negative end over here, it will repel. So that's why it will move, and will move towards the positive side. So you have two electrodes. One is cathode, and cathode, children, it is positively charged. Okay. And cations, cations are positive. Anions are negative. So anode is positive. Okay. So your DNA fragments will be moving towards the anode. Now you cannot see the DNA fragments really easily. Even though they are getting separated, you cannot see them with the naked eye. So what do, children? You basically treat them with something that is known as ethidium bromide. It's a very important question. Okay. So many times it has come. Ethidium bromide. What does this ethidium bromide do? Ethidium bromide is a chemical that goes and gets into the DNA fragments in between the bases and stuff like that. If you put in more quantity, it might manipulate your DNA. So you have to be very careful with that. But yes, ethidium bromide is there. Now, ethidium bromide, after putting ethidium bromide also, you won't be able to see it. Okay, so what do you need after that? You need to view it under UV radiation. You need to view it under UV radiation. At this point of time, you will find a strong orange color, a dark orange color. Now you have to remember the color, you have to remember UV radiation, you have to remember ethidium bromide. But if you remember this, probably you can solve lots and lots of questions. Okay, so it is ethidium bromide in UV radiation that becomes the correct answer. This came in NEET 2020. So many questions from NEET 2020. You see how many questions have come from this chapter. I have not picked all of them, but yeah. Still. Now next question. Following statements describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. You have to find the incorrect statement. So the enzyme recognizes a specific palindromic nucleotide sequence in the DNA. We know that. It definitely recognizes a palindromic sequence. So this is the correct. This is correct. Okay, we have to find incorrect. The enzyme cuts DNA molecule at identified position within the DNA. Yes, it can. Because it's a restriction endonuclease, it can cut within the DNA, so that is also fine. The enzyme binds DNA at specific sites and cut only one of the two strands. Was it true? Is it true? Not was it true? Is it true? Over here, you saw that this is a strand. It was making cut in both the strands parallelly, right? So obviously this is not true. Over here, it is said that they are making cut only in one particular strand, one of the two strands. So this becomes wrong, children. So this is your answer. We had to find incorrect one. This is the answer, but still let's read the other one. The enzyme cuts the sugar phosphate backbone at specific sites on each strand. Yes, it definitely cuts in the sugar phosphate backbone. So your DNA is made up of sugar and phosphate backbone. So it is cut there, which is why it is being separated. So that is also correct. This becomes the wrong answer. So always when you're asked to find incorrect options and correct options, sometimes the question, there might be something that might be correct in a question. So you have to go ahead and read all the options. Please do not get stuck at one particular thing. Please go ahead and read the whole question and then only go for the answers. Now, question number 11th. Given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. DNA is negatively charged molecule and so it is loaded on gel towards the anode terminal. So it is negatively charged. So is it loaded near the anode terminal? So this is the anode, this is the cathode. Where will I load my DNA initially? I'll put it near. Okay, I did something. Wait. I will put it near cathode. If I put it near cathode, it will repel because DNA is also negatively charged. It will repel and go towards that side. So that statement is wrong. Okay. What do you have to incorrect statement? So this is wrong. Okay, agreed. Now DNA fragments travel along the surface of the gel whose concentration does not affect the movement of the DNA. Now this is one extra information I told you in the class when I spoke about gel electrophoresis, but still. So in gel electrophoresis, you feel that the DNA is moving on the surface, but it's not like that. The gel is basically showing a sieving effect. Okay, what does it show, children? Sieving effect. That means inside within the gel it is passing through. It is not passing through the surface. It's not passing through the surface. This becomes wrong. Okay, so this statement is also wrong. Again, a very small information. Okay, so in your book, in your NCET textbook, they have mentioned about the sieving effect, but they didn't tell about whether they are moving in the surface because they thought it's understandable. So that is the thing. That is the thing. Sometimes we have to go read our NCET, and sometimes when we do not understand it, we have to seek help. We have to use your ChatGPT or whatever you have in source and just go through it and find the thing. Okay, because questions can come like that. Now, smaller the size of DNA fragment, larger is the distance it travels through it. It is correct, children. Smaller the size of DNA, obviously it can move, small things can move really fast, okay. The bigger things, the heavier things, they stay back. So it moves farther, it travels through it, is correct. Pure DNA can be visualized directly by exposing UV radiation, children. Do you think it can be directly viewed under UV radiation? No, you need ethidium bromide for that, okay. So this becomes wrong. And you had to find the incorrect statements. What all are incorrect? 1, 2 and 4. 1, 2 and 4. This becomes the answer. Okay, cool. Are we understanding things? I hope we are understanding things. I am trying to explain it a bit more for children who have not went through any classes of mine. Okay, so which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocyte? Now, human word is coming over here, children. And you have studied about different vectors. You have studied about different vectors. Vectors as in the part that will be carrying my gene of interest. Okay, so basically it can be an organism also. Okay, so in the case of plants, the most common vector that I am having is Agrobacterium. tumefacients. Okay. In the case of bacteria and stuff like that, I have bacteriophage, I have certain other plasmids and things like that. Okay. Now, yeah. Now, in the case of human beings, I have retroviruses. For example, HIV is a retroviruses. So, they are the one that can be used. So, over here, the TI plasmid comes from Agrobacterium tumefaciens. The alpha phage, this is phage, sorry, not alpha phage. Basically, this phage that you see over here, talking about bacteriophages, things like that, PBR322 is again a vector, but you don't need them in human lymphocytes. Okay, so basically, it's used in bacteria and stuff like that. So, this retrovirus thingy that you see over here becomes the correct answer. Please have a note of these things. This question would come regularly. Okay, this came in NEET 2018. So, what do you mean by this question is that I can alter the DNA over here and I can use this organism to pass my DNA into plants. So, this is Agrobacterium tumefaciens. It's a very important bacteria. It causes trouble actually, but I am taking the troublesome thing out and I am putting my gene of interest and making it infect the plant because I want to pass the gene of interest there. Similarly, bacteriophage and things are also like that. Retrovirus in the case of human beings are also like that. Okay. Now, 13th question. What is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? Okay. What is the criteria for DNA fragments movement? What controls the DNA fragment movements, or what are the things that you should be knowing about it? The smaller the fragment size, the farther it moves. It is true. Okay, the smaller the fragment size, the farther it will be moving. Farther as in more distant it will be moving. Okay, positively charged fragments move to farther end. Do you think positively charged fragments, who are the positively charged fragments? We do not have any positively charged fragments. DNA itself is negatively charged. Okay, now negatively charged fragments do not move. Is that true? No, they will move towards the positive side. Now, the larger the fragment size, the farther it moves. The larger, do you think large fragments would be moving farther? No, children, they will be moving really slow. So, this becomes the correct answer. This came in NEET 2017. Cool. Now, going ahead with the next question. The introduction of tDNA in the plants involve what? Exposing the, exposing the plants. So first thing, let me explain the question. Okay, introduction of tDNA. What does tDNA stands for? tDNA stands for transfer DNA. A little brief about it. Transfer DNA is found in Agrobacterium tumefaciens. So, introduction of transfer DNA into the plants involves, and if I want to put this transfer DNA into the plant cell, then what has to be done? Exposing the plants to cold for a brief period, allowing the plant roots to stand in water, infection of the plant by Agrobacterium tumefaciens, because Agrobacterium tumefaciens, children, it has the plasmid which is known as TI plasmid. TI plasmid stands for tumor inducing plasmid. And how does it induce tumor, children? Because it have a very specific DNA sequence which is known as tDNA or transfer DNA. Okay, it is known as tDNA or transfer DNA. So, when this naturally they go and infect the plant cells, they bring out certain infections, then they cause a disease which is known as crown gall disease. It's cancer, it's a tumor that is being formed. What do you call it? Crown gall disease. Now, how do we use them as a vector? Is that we remove the tDNA part. Okay, we remove the tDNA part, and instead of tDNA, we are putting what, children? Gene of interest. So, similarly, they will go and infect, but they do not know that they do not have the tDNA. Instead, they are putting the gene of interest. That's how things work. So, they are not talking about gene of interest thing. They are asking you how does the normal tDNA introduction take place in the case of plants. So, only if it is infected by Agrobacterium tumefaciens, then only it can get the tDNA. So, infection of plants by Agrobacterium tumefaciens is the answer. Okay. So, do not get confused, like there's a thing that how do we put our gene of interest in the host cell by electroporation, by heat shock treatment. Don't go in that particular line. They specifically are talking about tDNA, so it has to be Agrobacterium tumefaciens. So, infection of plants by Agrobacterium tumefaciens is the correct answer. This came in NEET 2015. Cool. Now, let's go ahead with the next question. Okay. Which vector can clone only a small fragment of DNA? So, you have different vectors. I told you about plasmid. Okay, I told you about plasmid, I told you about bacteriophage, I told you about retroviruses, this and that. Apart from that, you have bacterial artificial chromosome, which is known as BAC, and you have yeast artificial chromosome, which is known as YAC. Let me tell you a thing about it. Both of them, they are artificially created. It's not naturally occurring. They go and infect the they are inserted in the bacteria. It is inserted inside the yeast, okay, whenever we want to put a gene of interest. And the very unique thing about them is that these are the chromosomes which basically can accommodate a larger, okay, a larger piece of gene of interest. Okay, but usually the plasmids and everything, if you talk about plasmids, they have short fragments of DNA that can be inserted. So plasmid, if you talk about the ranking, okay, if you talk about, this is the first rank, this is the second rank, and this is the third rank. This is first, this is second, and this is third. I made it little top. Right. Okay, so first will be obviously BAC and YAC, larger fragments. Then you have cosmids, children. Okay, then you have plasmids. So plasmid is the one that carries the most smallest, let's say, fragment of DNA. So this becomes the answer. Cool, you can remember this, it's important again. So all this chotu information, no, questions comes around that, so you should be knowing about it. Now the colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of what? Okay, a little backstory of it, okay. So what happened is that I had a plasmid like this, okay, and over here, this plasmid's name is PUC8, okay. There is a gene for antibiotic resistance, ampicillin resistant gene, okay. What is it? Antibiotic resistant gene. For example, if I treat this particular organism who has this plasmid with ampicillin or something like that, it will still survive because it has the ampicillin resistant gene over there. Now, it also has one more thing. This gene is a structural gene, it is known as LAG-Z gene. And LAG-Z gene codes for something that is known as beta-galactosidase. This is an enzyme that is used for breaking down the lactose, okay. Now beta-galactosidase, no, this is a normal thing. This is how normally things work there, okay. Now in this medium of the bacteria that is having PUC8, okay, and all these things, if I am putting a substance which is known as X-gal, it's a chromogenic substance. What do you call it? Chromogenic substance, as in the one that gives color when broken down. So when X-gal is being put, what happens is that this beta-galactosidase goes and act on it and gives color, that is blue color. So this is a story of a non-recombinant plasmid. This is a story of a non-recombinant plasmid. Here everything is kind of normal. It happens like that. So they will be giving blue color colonies. Now what do? I have another plasmid which is a recombinant one. Over here I have the ampicillin resistant gene for sure. So everything is cool there. But instead of the LAG-Z gene, I am putting my gene of interest. I am putting my gene of interest there. So will it produce beta-galactosidase? No. So even if I put X-gal over there in this particular medium, having PUC8 plasmid vector like this, which is a recombinant, will it still give me blue color, children? No. Even if I put X-gal, there is no beta-galactosidase, and hence the color, there won't be any color change. It will be colorless colony. It will be colorless colony of bacteria. Here, since LAG-Z gene was working, beta-galactosidase was being produced, it was going and reacting with X-gal to produce a blue color. So non-recombinant ones are giving blue color, whereas the recombinant ones are not giving the blue color. They are giving colorless colony. I hope you understood the basic. And this process, no, where you are inserting a gene of interest, which is why the other gene is getting inactivated. Now the gene is inactivated, no, the beta-galactosidase is not being produced. You call this process as insertional inactivation. Insertional inactivation. You are inserting something and you are inactivating something. Got it? This is a basic funda. Now let's read it. So the question is, the colonies of recombinant bacteria appear white in color. The colonies of recombinant ones appear white in color, they are colorless basically, in contrast to the blue colonies of the non-recombinant bacteria. Because of what reason? Insertional inactivation of alpha galactosidase in the recombinant bacteria. No children, there is nothing like alpha galactosidase that we have studied as of now. It is beta galactosidase. Inactivation of galactosidase enzyme in recombinant bacteria. What is galactose? This is exactly wrong. Again, this is nothing that we have learned from. Non-recombinant bacteria containing beta galactosidase. Non-recombinant bacteria is containing beta galactosidase, which is why you are getting blue color colonies. This is correct. Insertional inactivation of alpha galactosidase. Again, it is wrong. So it is just because this contrast things is because the non-recombinant bacteria is containing beta galactosidase, or if the option was the recombinant bacteria does not contain beta galactosidase, then also it would have been correct. So this becomes the answer. If you are not able to understand this, pause the video for a second and go through it once more. You will understand. It's an easy thing. The first time when you hear it, it becomes a lot more difficult. I understand you as well. Yeah? Cool? Chalo, next question. Question number 17. The given figure in the is the diagrammatic representation of E. coli. Okay, it's not coil. E. coli vector PBR 322. Which one of the given options correctly identifies its certain components? So ORI is original restriction enzyme. No children. ORI basically which is there, it is the origin of replication. So this is wrong. ROP, reduced osmotic pressure. No, it is repressor protein, something like that. Basically, this is useful for, again, replication itself. For initiation of replication, you need ROP, which is right here. Okay, so that is also wrong. HIND3 and EcoR1 are selectable markers. HIND3 and EcoR1 are not selectable markers, children. Okay, the sites for them, they are not selectable markers. What do you have as selectable markers, children? You have your ampicillin resistant gene and you have tetracycline resistant gene, which is a selectable marker. What is a selectable marker? For example, basically I have a plasmid, okay, and I have ampicillin resistant gene over here and I have tetracycline resistant gene over here. Now what, usually they are resistant. So this particular plasmid will be resistant, or this particular bacteria having the plasmid will be resistant to both ampicillin and tetracycline. Now what do I do? Instead of tetracycline gene, I am putting my gene of interest and then I am treating it with tetracycline. Do you think it will survive? It won't survive because it has the gene of interest. So in this way, I am able to select this particular thing, that this has my gene of interest, which is why I call these particular antibiotic resistant gene as selectable markers. I call it as, children, selectable markers. So one selectable marker, you saw it over here also in this question also. Basically the LAG-Z gene was acting as a selectable marker. Ampicillin resistant gene was also acting like a selectable marker. But over here, HIND3, EcoR1, they are not selectable markers. Basically, they are enzymes which will cut over here in the recognition sites. So this becomes wrong. So ampicillin resistant, tetracycline resistance is antibiotic resistant genes. That is entirely correct. So this becomes the answer. Answer number 17th. Cool? Works? Now, next question. 18th, biolistics is suitable for. Biolistics means gene gun. Now this I taught it previously, right? So always remember, they might not give you an option like this in bracket. They might just ask you biolistic is used suitable for. Now biolistic, you think of it as a process and used in some other thing, okay, but it is just gene gun. And what do you mean by gene gun? Gene gun is used in the case of plants, okay. And what do you use in the case of animals? Micro injection. To pass down the DNA in the case of animals, you use micro injection. Cool? So basically, what am I actually doing? Whenever I need vectorless things for transferring the DNA, I don't need a vector to transfer DNA. What do I use? I use a gene gun there, which basically there's a gun sort of thing, not the gun that you're thinking of, okay. So over here, I am planting my DNA around microparticles of gold and tungsten. This is again a very important piece of information. Okay, microparticles of gold and tungsten is there, and I am coating it with DNA of my interest. I am coating it with DNA of my interest. And with high velocity, I am making it put inside the plant cell's nucleus. Inside the nucleus, I am putting it. Here, if I put, there will be enzymes like nucleases which would cut the DNA off. I don't want that. So basically, I am directly putting it inside the nucleus. This is done in the case of plants, and you call this as gene gun. Okay. In the case of animals, you use micro injection, where you have a small little thing, Okay, and you're using the micropipette which is there, you're inserting the DNA of interest. Okay, so please remember, for plants it's okay, they're strong, so they're okay with the gene gun. For animals, we cry with injections. No, we cry with gun, obvious. We cry with injection, so we cannot think of gun. So we have micro injections. So you can think in that way and remember it. Remember, gene gun is also known as biolistics. That is all the more important. So is suitable for what? Disarming pathogen vectors? No, children. Transformation of plant cells? Yes, children, this is the answer. Construction of recombinant DNA by joining with the vectors, DNA fingerprinting, nothing, just the plant cells. If you know about this, you get this answer correctly. Okay, next question. Agarose extracted from seaweeds is used in? Children, so we studied about gel electrophoresis. Why is the name gel? Because we are using a gel there, and this gel is known as agarose gel. And agarose gel is taken from the seaweed, my dear children. So what is this process known as? Gel electrophoresis. This becomes the correct answer. This came in NEET 2011. So champions, I want to take a break and say that as of now, while I was solving the question and you found that there were so many wrong answers, okay, and I couldn't do so much correct answers, what I studied is not enough, then I want to remind you, trust the process, remain calm, you will be able to do it. This is a trial thing, you are trying it out, and sometimes you fail, it's okay. Sometimes you fail again, it's okay. But you have to keep trying, because anyways you are going to give the exam, then why not do everything today itself? Whatever is possible, do it today itself. Give your best today, okay? That's all the more important. So this is the answer. Now next question. Question number 20. Which one of the following is used as a vector for cloning genes in higher organisms? Which one of them is used as a vector for cloning genes into higher organisms? So higher organisms means you're talking about animals basically, or human beings basically. For that you need retrovirus. We discussed this. The question is twisted again. Okay, so this becomes the correct answer. You didn't have to go through all these things. Direct retrovirus becomes the answer. Question number 20, my dear children. 21. Which of the following is commonly used in transfer of foreign DNA into crop plants? Again, it's a plant, and for plants I told you there are certain vector. So there is a vector which is known as agrobacterium. Tumor efficient, which has the Ti plasmid, which has the T DNA. You cut out the T DNA and you insert your gene of interest and then you put it again. Okay, so basically the answer for this question becomes agrobacterium tumefacciens. Works? Cool? Chalo. Next question. 22. Restriction endonuclease does what? Synthesizes the DNA, cuts the DNA molecule randomly, cuts the DNA molecule at specific sites, restricts the synthesis of DNA inside the nucleus, nothing. Cuts the DNA molecule at specific sites, specific palindromy sites. Okay? This becomes correct. Now, to confuse you, they can give you an option like this. Cuts the DNA molecule at specific sites, specific ends of the DNA. Okay, or cuts, that will be one option given to you. Second option will be cuts the DNA at specific sites within the DNA sequence. So you should be knowing it's within the DNA sequence. Okay, so give attention to this small details. My mouth, I'm kind of juggling around. My tongue is not working properly because of too much talking that I'm doing nowadays. Okay, so question number 23. The most thoroughly studied of the known bacteria-plant interaction is? Again, the same question, children. Cyanobacterial symbiosis with some aquatic ferns, gall formation on certain angiosperms by agrobacterium. I told you there's a disease which is being caused. It is known as crown gall. Now, if you do not know about crown gall, then you will be confused. I have studied about agrobacterium, but I do not know about crown gall. And angiosperms means flowering plants. Nodulation of sesbania stems by nitrogen fixing, plant growth stimulation by phosphate solubilizing bacteria. No. This is the answer. Okay, so every time you do the question practice, the next question becomes easy for you. So you have to practice that. This is 2004 question. Okay, going ahead. 24th question. Match the organism with its use in biotechnology. Bacillus thuringiensis, BT, you know, BT cotton and things like that. Okay, so they have cryproteins, which we have not studied as of now, but let's keep it like that. Thermus aquaticus, okay. gives us a polymerase which is known as Taq polymerase. T stands for thermus and AQ for aquaticus. So this is the bacteria that lives in very high temperature, very high temperature. So they have a DNA polymerase. This polymerase, no, Taq polymerase, we have given the name. It's nothing but DNA polymerase which is helping in DNA replication. Okay, in PCR and all, you use Taq polymerase for DNA replication because the temperature is maintained really high. So Thermus aquaticus is for DNA polymerase. Now, Agrobacterium tumefaciens, children, it is a cloning vector. It is a vector, right? I am putting my gene of interest and then I am making it use things like that. Salmonella typhimurium. Salmonella typhimurium is the first construction of first rDNA, that is recombinant DNA molecule, was made possible because of Salmonella typhimurium. Stanley Cohen and Herbert Boehm were responsible for this. In the initial part of the chapter, we discussed it. Okay, so Salmonella typhimurium is the first, was helping in the first recombinant DNA molecule, and along with it we were using E. coli also over there. Okay, so this is the answer. Let's see. Bacillus thuringiensis is fourth. Okay, A is with fourth, B is with 3. This is NEET 2020 question, my dear children. Works? Easy, no? Chalo. So even if you do not know about this, if you are able to match all these things that we had there, it is there in the chapter, you will be able to know the answer for this also. And so here only you see the B option is going with DNA polymerase, which is the, so nowhere the B option is going with the third one. Over here it's the answer that you already got. You already got the answer because you are very sure about Thermus aquaticus is a DNA polymerase. So you get the whole answer because here there is only one option which has 3 in B. So that's how you solve the question. 25th, the correct order of steps in polymerase chain reaction is. So polymerase chain reaction means you want to create multiple copies of the DNA, so many. It's a machine, okay? It's a machine that is being used. So over here the first step will be, I want to denature, I want to separate the DNA strands. So that is known as denaturation. And this is done at almost 99 degrees Celsius, 98 degrees Celsius, okay? Too much high temperature is required for that, for cleaving them. Second step is that I would put primers over here in the strand. Okay, so that primer gives a start for the extension of the DNA, for replication. Okay, so for that I need primers, and primers are added over here. So this will be 3 dash, this will be 5 dash, this will be 3 dash, this will be 5 dash. Okay, this is known as annealing, children. So here temperature is again put down, it is 50 degrees Celsius, something like that. And then again, after annealing, what do you need? You need extension. The next step is extension, where you are extending the DNA. So over here you have the DNA polymerase and you need a temperature around 70 degrees Celsius. Again, you're increasing temperature. So it is denaturation, denaturation, annealing, and then extension. This is the answer. And these things that you see over here, they are nothing but primers. So addition of primer for DNA extension is required. Okay, DNA polymerase cannot randomly go and start like that. It needs a backbone there and then only it will start. So that's why we need a primer over here, which is known as annealing. Cool, works. Let's go with the next question. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? So why do you need to degrade the enzyme? First thing, if I want my gene of interest, or probably if I want a plasmid from an organism, what do? Basically, I have to break open the outermost layer. So in the case of plants, you have cell wall, okay, and the cell wall is made up of cellulose. So you need an enzyme which is known as cellulase for breaking it down. Okay. Now in the case of fungi, again it also has cell wall, and this time the cell wall is made up of something that is known as chitin. So you need chitinase. Then you talk about, this is first, this is second, then you talk about bacteria. Third, okay, it also has a cell wall made up of peptidoglycan and stuff like that, and you need an enzyme which is known as lysozyme. lysozyme. And the interesting fact is that in our tears we have lysozyme, in our saliva we have lysozyme, and this lysozyme, that's why we tear up when something comes in. It's basically, it's killing all the pathogens, so all the bacteria and everything will die off. Obviously, if they not have the cell wall properly, if it is lysozyme is too much, it will kill the whole thing, right? So that is why we have that. So anyways, as of now we are not planning to kill them, we just want to cleave their outermost part so that we can isolate the DNA of interest, or basically isolate the plasmid of interest, whatever is it. So basically for isolation of DNA we are doing this. So these are the enzymes that is written. So fungi for chitinase is correct, bacteria lysozyme, plant cell cellulose, all of correct. But this, if you see, it's not mentioned, it's wrong. Okay, it will be wrong, my dear children. This came in NEET 2013. Okay, this is the 26th question that we have. Now going ahead, 27th. The figure below shows the 3 steps A, B and C of polymerase chain reaction. Select the option giving correct identification together with what it represents. So over here you see this is a DNA strand, and this region I want to amplify it. So for that, first thing that I do is, they have skipped one thing. So this is A, this is B, where I am putting the primers, and this is C, where I have extended everything. Okay, now we have to see what is correct. So B is showing denaturation at a temperature of about 98 degrees Celsius, separating the 2 DNA strands. No, children, it is not showing denaturation over there. Here you see the primers are also there. So it has moved to the next step, that is annealing. So again, A figure shows denaturation at a temperature of about 50 degrees Celsius. No, there is no denaturation, they are still together. And 50 degrees Celsius is again wrong. Denaturation is done at 98 degrees Celsius. Now C shows extension the presence of heat stable DNA polymerase. Extension in the presence of heat stable DNA polymerase. Children, if you see, it has already extended itself. The complete extension is being done. Now if you talk about A, annealing with 2 sets of, A is, sorry. If you see this option, C is extension in the presence of heat stable DNA polymerase. Yes, so that suits best, but still we will see this also. In option number A, what do you have? Now option number D, A is annealing with 2 sets of primers. No, children, it is B where you are annealing things. So the correct option becomes this. Okay, this came in May 2012. 27th question. So if you're not aware about denaturation, what temperature takes place, or annealing or extension, you're not aware about the picture, you won't be able to answer this. You have to go through things. Okay. Next question. Stirred tank bioreactors have been designed for. So stirred tank bioreactor as in, so there is a bioreactor like this where you have a machine, agitator machine, which has blades which will be stirring the whole content there, and you will be having a pump for air to get in. Okay, you will be having a pump for fixed amount of nutrients, or basically just nutrients. You're putting nutrients, you're putting the medium and everything there, and it is basically mixing thing. So this is a stirred tank bioreactor. With this information, I want to tell you one more thing. There are 2 types of stirred tank. One is simple, which I have drawn over here. One more that is sparged. Okay, what do you call it? Sparged stirred tank bioreactors. So what happens over here, they are also mixing the air properly, but in the case of sparged, no, the air comes from air bubbles like this, and which helps in good mixing. So sparged is better because their air comes in the form of air bubbles, and it will gradually mix really well. So anyways, why do we need stirred reactor? The first place is that we need to mix the air efficiently. Obviously nutrients also you have to mix, but you want to make sure that they are not doing anaerobic, the bacteria and everything which is put there, they are not doing anaerobic respiration, but they are doing aerobic respiration. We want them to do aerobic respiration. I feel like I'm talking in Arabic. Okay, I think I didn't have breakfast. So yes, so aerobic respiration is needed over here. We do not want them to do anaerobic respiration, which is why we want availability of oxygen really well throughout the process. So that is why we have stirred tank bioreactors. Don't go for anything else. Nothing related to nutrients, nothing related to anything, because this is given in your NCERT. Even though we want to mix the nutrients also, some bacteria might be crying for nutrients. I want nutrients. I won't be growing without nutrients. Some might not. So basically because they already have it. So it's for mixing the nutrients as well. But in your NCERT clearly they have mentioned for good availability of air, you need stirred tank bioreactors. So we have to go with that. So addition of preservatives to the product, no. Purification of the product, no. Ensuring anaerobic conditions in the culture vessel, no. Availability of oxygen throughout the process, my dear children, this is the answer. 2010, the question has come. Easy question, no? And it's fun also. Okay, now next question. So will you remember that sparged, okay, I'll write properly if you want. Okay, just a second. I know when I write fast, my handwriting becomes all the more terrible, which I don't think it is that terrible. We have simple stirred tank bioreactor, and we have sparged stirred tank bioreactor. We have two things. So I say sparged is better, okay, because it gives out the air in the form of air bubbles. So over here gives out the air in the form of air bubbles, so it makes really nice. Cool, works. Now the next question, 29th. An analysis of chromosomal DNA using the southern hybridization technique does not use. So basically what happens is that it's not like okay, so you had gel electrophoresis being done. Okay, your DNA got separated. This is smaller fragments, these are the larger fragments. Okay, now I have to view it, right? For that, what do I do is that I have a nitrocellulose sheet. Okay, I just stick it like that and I take it. So my DNA fragments are now on the nitrocellulose sheet. This is the nitrocellulose sheet. Okay, and I have, oh sorry, the DNA fragments over here, because so many reactions cannot take place over there. Now I treat it with ethidium bromide or whatever, okay. I can treat it with some chemicals and then I can view it under UV radiation. That is, auto radiography can be done. This process where I am putting UV radiation, you call it what? Auto radiography. To view them properly, to view the DNA strands. So yes, in southern hybridization technique, I need electrophoresis to happen. I need bloating where I am taking a paper and bloating things down. I need auto radiography being done over there for viewing things. PCR, polymerase chain reaction, is not needed over here. So this is something that is not of any use in the case of southern hybridization technique. So this question is given in this particular chapter, where they have not mentioned about southern hybridization technique. So you should be knowing that this is how things work. You cannot randomly put ethidium bromide there and try and view it. Okay, obviously it will also react with a lot of things, but here it's all safe because you just have the DNA. Cool. And this process is known as southern hybridization technique. Works. Okay. PCR is done when you want to, for example, you have a gene of interest. Okay, now you want to amplify this in too much amount. That's when you use PCR. You want to amplify it. That's, for example, I got the DNA fragments which are smaller in size. I took this out and now I want to increase their clones. So at that point of time, I take the DNA strand and I put in PCR machine and I ask it to clone. Okay, to make multiple copies of it. That's when I need it, PCR. Next thing, the last question. Which one is a true statement regarding DNA polymerase used in PCR? It is used to ligate introduced DNA in the recipient cell? No. What is the use of DNA polymerase is being asked. So DNA polymerase, children, it is for DNA it is used for DNA replication, you know that. Okay, that is one very important thing. Fine. Now, in the case of PCR, there is a unique thing about this particular DNA polymerase. First unique thing is that I have a different name for it. What is the name, children? It is known as TAC polymerase. It is known as TAC polymerase, where T stands for thermus and AQ stands for aquaticus. Okay, so it is extracted from the bacteria, that is Thermus aquaticus, which stays in very high temperature areas. Okay, so I took the DNA polymerase. It is able to withstand high temperature, and with the help of it I am doing DNA replication, because this is a process where I am using too much amount of high temperature. Okay, now it is used to ligate introduce DNA. It is not used for ligating. Ligating means you want to fix the DNAs together. That is what you mean by ligation. So that for that you need DNA ligase, not this. It serves as a selectable marker. No, children, it has nothing to do with a selectable marker. It is isolated from a virus. No, it is isolated from a bacteria, Thermus aquaticus. Okay, then you have it remains active at high temperature. This is the answer. It remains active at high temperature. 99 degrees Celsius, 70 degrees Celsius, things like that. Okay, 70 degrees Celsius actually works. So these are the 30 questions. I hope you understood each of the questions, and still children, if you have any doubt, please feel very free to ask me in the comment section. I read your comments. I say thank you when I see good comments. I feel very happy when I see good comments. I feel equally happy when I see comments asking for doubts and stuff like that. So you can generally ask me any sort of doubts that you are having in the comment section. I will be there to help you out if you ask for help. Okay, so that's one very important thing. Something that's something that I can do. I'll definitely do it. Okay, and I hope this was helpful. Please make sure you're using all the mock test series and you are doing it by yourself. Whatever we have done in past, we have 9 mock tests being done already for all the different chapters. You can use it, you can try the questions out. These are all previous year questions. It would help you score really well. Okay, and in all of them I have explained like this only. So yes, that's all my dear little cuties. So if you think that this particular video was helpful for you, then do not forget to subscribe to our channel, that is Diksha Karnataka, and I wish you all the luck. Please don't feel sad for anything if you have not scored marks. It's totally fine. You are trying, and that means a lot to me. So bye bye children, take care.